Hi everybody. This video will serve as an introduction to Energene's visualization system and to our multi-genome capability in this visualization system. This system is based on an IGV viewer from the Broad Institute, which we have significantly improved. One thing we did improve is add tabs. So the IGV is basically showing on this part the reference genome, which is pointed here, in this case, a B73 maize reference genome. This point here, red dot, is showing the area in which we are zooming in here. So the whole screen here showing this part of the whole genome, and the last part of the screen is broken up into tracks. Each track is showing different information. In this case, this part of the screen, the track here, is showing alignments between reads and the reference genome, and this track here is showing other types of information, which is the scaffolds of okay. So here we will see scaffolds, which are the results of the specific iodine line de novo process, again aligning to the genome, and the IGV is using color coding to point out the differences between the sequence of the read or the scaffold to the reference gene. This part here is showing in another type of track the information of genes. So what we did is split the IGV visualization into tabs, and each tab is showing a different type of information for a specific region. So this tab here, for example, will show results of polymorphism analysis. Again, as you zoom out, you see more and more types of information, and so this tab is broken into many more small tracks, and so on. Another interesting feature of our version of the IGV is the ability to switch between reference genomes. So if we, again, switch tabs and we see, for example, QTLs, so these QTLs are mapped to the B73 genome, okay? If we focus on a specific area, and we can switch to another reference genome, in this case, an iodine genome, and we can see that the sizes of the physical genome in this area are different. So this is the iodine pointed out here, and this is the pseudo chromosome of the iodine, which is the results of our de novo assembly. And going back to the B73, you would see that the region which is on the iodine is 5,340 kb is now different. Okay, going back into the iodine, you can also see there are some differences in the annotation, and in this case there is an additional gene which is a PAV, meaning it's existing only on the iodine genome and not appearing on the B73 reference gene. Another nice feature is the ability to use genetic maps. So if we, for example, want to understand what is the centimorgan size of this area, we can go to a physical map, which shows a physical size, but we can also go to a genetic map, which shows the genetic distances. In this case, this is the IBM map coming from the public. And then we'll be able to see QTLs that are maps on the genetic map versus QTLs that are mapped only on the physical map. So the markers could actually not be fully mapped to the physical map, but we can integrate the genetic map and the physical map and enable the user to work with both.